My name is John Jakisik, and I, along with Ed Melanson, chaired the development of the Energy Balance World Congress. With that being said, I'm very honored and thrilled to introduce our first speaker. I know when I got started in the field, Dr. Bouchard was the one person, one of many, but one of the many people that I cited many, many times as I went through my training. So I'm thrilled to be up here to introduce him today. Our first speaker is Dr. Claude Bouchard. He finished, I won't go through this long list of credentials. I think most of us know Dr. Bouchard. But as we're speaking here this morning, he has over 1,100 publications and counting. So that's a lot of publications. Many of them have been very, very impactful in the field. And we see him as a leader in this area of energy balance, metabolism, genetics, and physical activity, and has done seminal work across all of these areas. He's currently at the Pennington Biomedical Research Center. He got his postdoc at the University of Montreal, his Ph.D. at the University of Texas, and the list goes on and on. He's been honored in many ways, and he's going to speak to us this morning on the regulation of body weight, physics, physiology, and behavior. I'm thrilled to present to you Dr. Claude Bouchard.
Good morning. So my task is to provide an overview of the role of physics, physiology, and perhaps behavior if time permits. This is obviously a very complex topic, and since this is part of the basic science component of the program, the emphasis will be on basic science in my presentation. First, I have no conflict of interest to report, and I'm going to use this outline. First, a brief introduction, which I've labeled a high-altitude view, and then the concept of metabolic individuality will retain our attention for a while. This will bring me to the ongoing paradigm shift in our understanding of adipose cell biology, the white, beige, and brown adipose tissues, the role of a genetic predisposition and the ongoing changes that are occurring there, and in summary, what I believe is the common scenario for the common form of obesity. I'm going to try to convince you that there is an area which we have, in the early days, probably the 1950s and 60s, we thought that this was a productive field of research for our understanding of obesity. And then it went into oblivion for a while, but our shifting understanding of metabolic individuality, I think, provides us a foundation for a new approach, a new look at the role of energy metabolism and the role it can play in the predisposition to obesity. And as Lavoisier said, life is a combustion. So the main drivers of this epidemic that we are experiencing around the world are to be found in the physics, in the physiology, and of course in behavior and environment. And behavior and environment are the major drivers of the big changes that we see around the world. But physics and physiology still account for a large fraction of the human variability that we see in the risk of obesity. It accounts for the fact that some people are very much more at risk than others, no matter the living circumstances. So I will emphasize those. We'll begin with the first law of thermodynamics. The increase in energy in a closed system, according to the law, is equal to the heat supplied to the system. In our case, in our system, the human body, it's dietary calories. minus the work done by the system, which, of course, here is energy expenditure. So we know from the first law that energy cannot change, cannot be created or destroyed, but it can be modified, it can change form. Thus, a weight gain, a gain in energy, therefore, occurs in the presence of increased caloric intake and or decreased energy expenditure. So the first law is really a conservation of energy law. It has been labeled as a bookkeeping law. However, we need to interpret the first law in light of the second one. And the second one is a dissipation law. And in the process of converting energy in the human body, which is not a perfectly efficient system, we have energy that is lost in a way that we cannot recover it in the body. And this is the case where we have losses that are found in the form of heat or excreted energy. So the central issue here, therefore, is to understand how inefficient is the human body at converting energy, and what are the sites where these inefficiencies occur. So both laws have implications for the regulation of body weight. Now, even though the human body is not a completely efficient system, you expect that the first law is approximately correct. That is, energy intake is approximately equal to energy expenditure. But the second law tells us that the energy cost of transforming nutrients into metabolic energy or stored energy, that energy cost can vary within people over time, and of course even more among people. So let's try to understand this metabolic individuality. As I told you, for 50 years ago, this was an area where we thought we would find the answer to chronic positive energy balance, and then we lost ourselves there because we're dealing with differences in phenotype that are very subtle, and the common view was that they were very difficult to measure. And they're still very difficult to measure. But we understand a lot more about the biology and the underlying molecular mechanisms that can engender these slight variations in metabolic individuality. So let's open that chapter. The processing of food calories into metabolic fuel varies in efficiency, and this is determined by several factors: hormonal and physiological status. For example, insulin, glucorticoids, steroids, and other hormones influence the fate of nutrients through, for example, energy partitioning, or the profile of metabolic fuel oxidized or stored. Likewise, the metabolic state in the liver, in the muscle, and the adipose tissue play a big role on these processes. These differences can rarely be observed at the minute molecular level when they occur, but we see them if we look carefully in experimental situations or in life situations. For example, we would expect that if this was correct, that there would be, even among genetically identical members of a species, there would be differences because some of these subtle differences in metabolic efficiencies would have been acquired over time or would change from season to season or across age. And such differences will be even larger among outbred populations. So let me give you an example here. Let's start with inbred organisms. Such differences in efficiencies are observed even among genetically similar individuals. For instance, weight gain in inbred mouse strain when fed under identical conditions and kept under the same caging conditions. That's I'm going to show you a slide to that effect. But the differences are even larger among individuals of outbred organisms, which support the notion of variability in the genetic predisposition as such. Here is an example here, done in the laboratory of Les Kozak when he was at Pennington. Here we have 112 male C57 mice inbred, raised under identical caging conditions. For a period of about 5 weeks, they were fed a normal chow diet, and then for 5 more weeks fed a high-fat diet. And as you can see, here at the end of the chow feeding period, there was between the low gainers of weight and high gainers of weight, a pretty large range, 6-7 grams, when the mean weight of the animals was about 23-24 grams. After the high-fat diet, these differences were even larger. There were some animals who were in the mid-20s, but some reached as high as 35 grams. There was no difference in locomotor activity in these animals, and the only way to explain this appears to be metabolic efficiency, but the site of the causation of these metabolic efficiencies, of course, is unknown. Similarly, another example, this time in human subjects, an experiment we did many, many years ago, 25 years ago, in which we overfed 12 pairs of identical twins with a standardized overfeeding protocol. 100 days of overfeeding, a caloric surplus of 84,000 calories per subject. 84,000, standardized. Composition, the macronutrient composition of the diet, also standardized. Well, we have, even though the weight gain is 8 kilograms, the standard deviations was 2 kilograms, a threefold range in weight gain between the low gainers and the high gainers. They were kept completely sedentary, fully monitored 24 hours a day, and studied only 4 pairs at a time so that we could control them even better. So an extraordinary range that cannot be explained by differences in caloric intake, cannot be explained by differences in energy expenditure of activity. So it has to be in metabolic efficiency. So what are the major sources of these metabolic individualities? Well, 5 sources will be discussed here. There may be others, and there are others, but I'm going to emphasize 5. Substrate cycling, coupling efficiency, calories lost in feces, uncoupling proteins and fat cells, and genetic differences. Let's take a peek at each of them. Of course, time will not permit me to expand too much, as you appreciate. So let's begin with substrate cycling. So when metabolic pathways run simultaneously in opposite directions, and they dissipate energy in the form of heat, that is, less product is generated and expected from simply the stoichiometry of ATP generated and consumed. That's when we have a substrate cycling. This concept was developed in the 1970s, mainly by Eric Newsom at the University of Oxford. And Eric, by the way, was a biochemist who became very interested in exercise and was a regular participant at these meetings here, and he gave talks regularly on the biochemistry of exercise. He was a superb biochemist. He identified many of these substrate cycles and referred to them as futile cycles and defined their relationship to energy balance. In a paper he published in the New England Journal of Medicine, he illustrated this point. So here's a hypothetical metabolic cycle. We have a substrate S and a product P, and when a typical cycle, only one step in the cycle. We have a conversion of A to B, but some of B is converted to P, the product, but at the same time, through the action of an enzyme 2, some of B is being reconverted to A. So part of the transformation from substrate to product is expended in a futile way because it does not generate a product as such. So that was pretty revolutionary, and he used as a typical example the fructose cis-phosphate and fructose biphosphate cycle in the pathway from glycogen to lactate. And we see exactly the pattern that I showed in the previous slide here. Now, he went further and identified about 25 of these substrate cycles. And for each of them, he defined where the steps were located and how they could contribute to variation in energy expenditure and metabolic individuality. So now we're 40 years later almost. What have we learned? Are these cycles rare or are they common? Do they generate large energy losses in the form of heat, not recovered as body mass? These are critical questions. They have been examined outside of the field of energy balance. They've been examined at the cellular level and in systems biology. So let me take you on a brief trip. I'm going to be inspired by this paper here, which was reported in the FACSM journal a couple of years ago, in which a whole metabolic network approach was taken. So not just looking at some cycles, but all the cycles in all the cells. And to do that, the group used a whole cell model of human metabolism as the main resource, combined with tissue-specific cycles using gene expression data. So they use resources available in the omics world, and they are located through bioinformatics and pathway analysis, all the cycles that have the potential to generate excessive energy expenditure compared to the expected products. They also look at the localization of these pathways, but I will not get into this here. They identified about 100,000 substrate cycles with evidence of futile or excess energy consumption. Now, they also found that a lot of them were short cycles with 1 step, 2 step, 3 steps. But also they identified several that were multi-stages, large pathways. Some, for example, 587 at 6 reactions, more than 2,000 at 10 reactions, more than 2,000 at 40 reactions in them. So this seems to be ubiquitous in nature. Here is the distribution of the length of those cycles. On the x-axis, we have the length, the number of steps in the futile cycles. And as you can see, they had some even at about 100 different steps. But the majority were closer to the 20 to 30 steps. They also asked the question, how much energy is expended in a futile way in these cycles? So here's the distribution of the frequency of the excessive ATP consumption. And as you can see, it appears that there's been a selection for a very low inefficiency. Most of these cycles use an excess of ATP that's very small. But look at the number of them, though. We have 5,000, 6,000 of those cycles. And you have some that are expending more futile energy. And, but in a way, it appears that the evolutionary pressure on our biology has been such that it favors low ATP wastage. So we're talking about a problem that is a very different dimension than Eric Newsom was proposing 25 years ago. We thought it was limited to some specific cycles in some pathways, but now it's ubiquitous in nature. It's the way our biology is. Some slight inefficiencies, and they probably vary depending on the conditions of the cell, influenced by hormones, influenced by adipokines, cytokines, myokines, etc. Now, coupling efficiency is another area where it's reasonable to expect that it might be a site of metabolic individuality. This refers typically to the coupling efficiency of mitochondrial oxidative phosphorylation to ATP generation. With poor coupling, the proton gradient is dissipated. Protons are leaking across the mitochondrial membrane without generating the ATP, without using the electro force to generate ATP. This is primarily in the mitochondria, but there are also potential other sites for that. For example, all the pumps that we have at membranes pumping ion against gradients. That's also another area, potential areas where we have metabolic inefficiency. Variation in metabolic efficiency, I should say. So Mary Ellen Harper and colleagues published this review of that problem a couple of years ago, several years ago, in Annual Review of Nutrition, and I've been inspired by their paper, their view of it. They look at all 4 stages of mitochondrial coupling respiration, looking for efficiency. And the bulk of the literature is saying us that, yes, for the stages 1 to stage 3, efficiency of coupling is quite high. It's not perfect. It's not 100%, but it is in the 90%. So there is some wastage there, some slippage, and we have some futility of energy expenditure there, but it is small. For stage 4, though, it's higher. The coupling efficiency is only of the order of 75%, so we have more leakage there. However, this is amplified dramatically in brown adipose tissue, where through the action of the uncoupling protein 1, UCP1, inefficiency, especially in response to beta-adrenergic receptor stimulation, there the leakage is really high. The proton leak is dramatic. And we have, of course, a lot of thermogenic potential in that particular tissue. But there is inefficiency even in the absence of uncoupling protein 1. Mitochondria does not generate energy at an efficiency of 100%. Then we have some calories lost in feces. This has been controversial. I remember we were discussing this 30, 40 years ago, and we thought that maybe a site, but a site of variation among people, but we could not generate data that would convincingly support that hypothesis. It's coming back, particularly with our a growing understanding of the role of the gut microbiome on metabolism. So the gut microbiome is necessary for the digestion of dietary polysaccharides that we cannot digest on our own. We don't have the proper enzymes for that. So the gut microbiome does the job for us. And these polysaccharides include the oligosaccharides and the resistant starch and the fructose and some others. But the microbiome also is critical for other things, to synthesize vitamins, some vitamins and cofactors. It regulates in part the motility and the vascularization of the GI tract, and it participates in the control of inflammation and immunity. So it is an evolving biology. Now, it is a complex organism that we have. It consists of up to 100 trillion microorganisms. Actually, 10 times, we have 10 times more cells in our gut than we have in the rest of our body. Collectively, all these microbial genomes exceed the number of genes that we have in our own human genome by a factor of 150. So we wonder which DNA is driving. And each of us, there are about 1,000 species of these microbes. Each of us is housing in the gut about 160 different types. Some are present in very small quantities, but there's a huge diversity there. There are lots of study on this. I'm going to focus on one issue. Here is an example of differences between obese and lean individuals in some of bacteria types. For example, those listed vis-a-vis the obese individuals, there are here about 10 different types, are not found on average in the microbiome population of the lean people. Conversely, these 5 types of bacteria are not found in the gut of the obese individuals. So we have differences in some of the subpopulations that are present in obese, not in the lean. And then we have differences in the proportion of the most prevalent microbiome in the obese versus lean as well. However, the important point is in this slide. In people, there have been estimates of the amount of energy lost in urine and feces, and they typically range from 3 to 10%. Varies depending on the conditions, the diet, the health status, etc. And thus far, it has been proven to be almost impossible to relate them to body weight and the regulation of body weight in people. But inpatient studies, now recent inpatient studies, suggest that lean individuals harvest less calories. In other words, they leave more calories in the gut than obese people do in response to overfeeding. That's an interesting development. In the overfeeding study that we did, that I showed you one slide of 25 years ago, we were able to measure the calorie content of the gut in 16 of the 24 subjects, and we saw a few things, but we could not relate that clearly to the amount of weight gain. But when you take lean and obese and you overfeed them, the lean retain less calories more calories in the gut than the obese. Now, importantly, the proof of concept is in the last point. Obese mice, obese mice in this case, harvest more calories from their daily intake. And this was shown very nicely in an experiment in which they took lean mice and remove all the bacteria in their gut. And they were made totally germ-free. And they transplanted the bacterial microbiome from the OB mice into the lean mice. And the lean mice began to retain more calories from the diet and gain weight more than the lean control. So the bacterial genome seems to have an important function, in a subtle function, not a large one, a subtle function in modulating body weight. And as people gain weight, the function of the microbiota changes. Okay. Also, a source of metabolic efficiency that we're beginning to understand, appreciate, and on which there's a lot of research going on at the moment, is our understanding that we don't have two populations of fat cells. We have three, and probably even more, multiple variation on the three phenotypes, the three major phenotypes. That is the white, the beige, and the brown fat cells. An adipose cell is, we understand now, we've understood this for about 20 years, is truly a secretory organ. It's an endocrine, exocrine, paracrine, and autocrine organ. And here are some of the excreted molecules. You can recognize adiponectin, insulin-like growth factor 1, leptin, resistin, TNF-alpha, vistatin, etc. And this is only a partial depiction of the number of excreted molecules. So that has a role to play. But in addition, we now are beginning to perceive that brown fat, which we thought was a characteristic of the newborn, and that over time in human beings, the brown fat would atrophy and would be inert. There have been attempts in the 60s and 70s and 80s to measure that, to try to stimulate brown fat with beta-3 agonists, and they were not successful. But for about 10 years now, we have new evidence from PET scan data that brown fat exists, and in most humans, it can be activated. It's more difficult in older people and in people who have metabolic anomalies such as type 2 diabetes, for example. It's more difficult to activate brown fat in them, but in general, in middle-aged people and young adults, it can be activated. But importantly, a new type of fat cell has been uncovered, and it has been labeled a number of ways, but we will use beige in this presentation. And conversion from some white fat cells can occur with the right stimulation, including exercise. Clearly, brown fat cells have the highest capacity for substrate oxidation. They are a thermogenic machine. They have an uncoupling protein 1 expressed at high levels, and they, upon beta-3 adrenergic stimulation, they produce a lot of energy. But some white fat cells can be converted and display almost the same metabolic properties as brown fat cells. They are metabolically reprogrammed, and they express all the thermogenic markers of brown fat cells. And among other things, the beige fat cell acquire in their conversion a higher capacity for fatty acid oxidation, which also diminishes plasma free fatty acid levels and diminishes fat deposition. So many papers have looked into this. This is one that you should look at if you are interested in published in the JCI last year. And here is what we understand at this time. I'm not sure if you can see all the details as if probably not. Let me try to summarize the essential points. Oops, sorry, that's my mistake. Let's begin with the brown adipocyte. The lab of Bruce Spiegelman has shown that the precursor of brown adipocytes are found in the mesodermal tissue, embryonic tissue. And these precursor cells are either destined to become muscle cell or brown fat cells, depending on the presence of transcription factors. To embark on the path of brown adipocyte, you absolutely need to have the transcription factor MYF5. And then once the brown adipocyte lineage, oh, I lost my pointer. Voila. When the lineage is acquired, it's sustained. On the other hand, for the white fat cells, also originates from mesodermal stem cells. And here, in the presence of appropriate environmental cues, such as cold, PPAR gamma agonists, or exercise, the cell acquires some properties that replicate the metabolic profile of a brown fat cells. The difference between those that acquire move along this lineage and the others who move in the direction of the brown fat cells is the absence of the MYF5 transcription factor. So this doesn't work anymore, John. Oh, it's back. No, it's erratic. So there are three paths that are currently, well, I'm sorry, I don't have a pointer and I don't have a mouse, so I cannot tell you. There are three paths, label 1, 2, and 3, Roman. And these three relate to the differentiation from beige to confirmed beige adipocytes, or in white precursors, whether they can be, once they have acquired the full white precursor state, whether they can be converted to beige, and whether a white adipocyte differentiated into a beige adipocyte can be reconverted back to a white adipocyte. So those are currently the attention of a lot of basic scientists in this area. But beige differentiation can be induced by chronic exposure to cold. There may be a time window for this, exercise or PPAR agonists. And it has begun to be looked at what in exercise can promote the beige adipocyte recruitment. And the candidate molecules are listed here, and it may be not one, it may be multiple molecules that are triggered, secreted, enhanced by regular exercise. One of them is beta-aminoisobutyric acid, beta-3 adrenergic stimulation, of course, FGF21, secreted from the liver or the muscle, interleukin-6, hyrisine, meteorin-like myokine secreted from the muscle, and vascular endothelial growth factor. Those are candidates at the moment for the role of exercise in recruiting a white adipocyte to transform them into a cell with a beige profile. Okay, so if we have subtle differences in the metabolic efficiency throughout all of our cells, you know, and multiple sites, and I confine my remarks to only a few today, some of the genetic predisposition that we see in human population, is it acting through modulation of metabolic efficiency? That we don't know, but that's a question that we can ask. So let me tell you where we stand in this area. The genetic component to obesity is still a matter of debate, but let's assume that it is about 50% of the age and gender adjusted human variation in risk of obesity or BMI level for that matter. The common and rare DNA variants that have been identified thus far relate mostly to the genes that are involved in the central regulation of appetite and satiety and energy balance in general. But thus their primary relevance appears to be to behavior, appetite, satiety, and maybe activity. However, many other variants have not without a clear function at the moment. They are associated with genes whose biological role we poorly understand, and therefore there is a potential there for a contribution to metabolic individuality. So we have a lot of information on now, thanks to the ENCODE project, on the complexity of metabolic regulation in the biology of a cell. ENCODE is the project. ENCODE is the encyclopedia of the non-coding elements of the genome. A consortium of dozens of laboratories around the world work together on this. Here are some of their conclusions. First of all, our human genome has about 21,000 protein coding genes. That's all, 21,000 protein coding genes. Now each of these coding sequence may encode for several peptides because they are splices, they have a number of initiation site, so it's not a single peptide that comes out of all of these 21,000. Actually, people think that these 21,000 protein genes may encode for more than 100,000 peptides. We have in the genome almost 3 million protein binding sites in our DNA. Sites where protein will bind and will influence the transcription, the expression of the genes, or the lack of expression, the repression even of the genes. Our genome encodes more than 8,000 small and more than 9,000 long non-coding RNAs, and these are involved in modulating transcription, translation, the life of the cells. And all these molecules interact with one another so that so many sites where we can have subtle differences in the energy demand among people. So we have 21,000 genes, and the drivers of these, the expression of these genes are the transcription factors. And we have more than 1,500 transcription factors. We don't know yet. There may be 100, 200 more, but that's the ballpark figure. We're very close to have identified them all. And these transcription factors bind at 8% of the genome, which means that on average, a transcription factor is involved in the expression of 15 genes. So when a transcription factor triggers the expression of a gene, it probably triggers the expression of several other genes at the same time. But very importantly, we have more than 70,000 promoter regions and 400,000 enhancer regions that are located in the vicinity or sometimes quite far away from the coding sequence of a gene. and those regulate the expression of the gene as well. In other words, we have about 23 of those modulating regions per gene. So think about that. You know, it's like an orchestra. Before a sound comes out, a gene is expressed. You have 100 musicians contributing to that sound. In this case, the expression of a gene. So many sites, so many levels where energy and metabolic efficiency can vary in a subtle way among people. In addition, in our genome, 3 billion base pairs of DNA, we have 40 million sites that are polymorphic with variants common in the population, meaning it's carried by at least 1% of the population, 40 million sites. Any one of us, anyone in the room, carries about 3 to 4 million of these variant sites. Excuse me. 3 to 4 million is enough to account for some of the differences among people, if they are at the right place, if they have metabolic implications. In addition, each and every one of us carries at least 200,000 and maybe as many as 500,000 what we call rare variants. And finally, large numbers of copies of variable sequences covering almost 10% of the genome exist as well. So it's not surprising that we have subtle differences in the predisposition to obesity, and it probably begins in this slide and the previous slide, in the phenomenon that I just described. Where do we stand in our understanding of the role that they play in obesity? Well, the most comprehensive of the papers is this one, published about a year ago in Nature. And what it did, it showed us that, first of all, it was based on the sample size of 339,000 people. So that is enough to create high p-values. But the genome was explored with millions of markers in each individual. And 97 single nucleotide polymorphisms were found to be associated with the BMI or with the risk of obesity. It accounted for about 3% of the variance in BMI. So we're far from the 50% that we understand is the irritability of the BMI. But those are highly significant, 10 to the minus 8. Now, it's 3% on average, but it's much more meaningful if you have a high risk profile over a low risk profile. In a situation like this, we have almost 200 alleles. Each person can have a maximum of 200 alleles, or 0 if you have no risk alleles. And in this particular population, if you had less than 78, and you compare those who were in that position, there were about 100 people who had less than 78. You compared them to those who had more than 104 risk alleles. The difference was 12 kilograms. That's important. Finally, based on the observation of that study, plus its concordance with the similar studies done in other samples, it was predicted that if we had a sample size of about 1 million, it would result in about 1,000 SNPs that are significant, and it will account for about 20 to 25% of the variance in BMI. So we're getting there slowly. But as you can see, the effect size is small. Thus far, in all the studies, 200 DNA variants have been identified. The genetic effect is there, but it's not a genetic determinism. It's a slight predisposition. And within these variants, many will undoubtedly be shown to contribute to metabolic individuality. In summary, physics is essential for our understanding of the regulation of body weight. Therefore, yes, energy balance is approximately equal to energy expenditure as we measure those. However, the human body is not a perfectly efficient machine, and metabolic individuality translated to differences in the predisposition to weight gain or weight loss under identical behavioral conditions. Physics is relevant, behavior is critical, but I submit that physiology also ranks high. And here, I did not cover all of the areas. I cover only those that are in the blue on the slide. But a scenario that I think is operating is something like the following. We have a number of factors. For example, being older is a risk factor. Being a female is a risk factor for having a slightly higher weight. Being an African American, being a non-smoker, coming from a low SCS, eating a lot of fast food, sugar, sweetened beverage, being sedentary, having a high respiratory exchange ratio, low leptin levels, metabolic efficiency 1, being very efficient at, say, 1 trait, low sympathetic nervous system activity, low growth hormone and IGF-1 status, low androgens, low skeletal muscle oxidative phosphorylation potential, metabolic efficiency 2, less calories lost in the feces, low brown adipose tissue activity, low conversion of white to beige, having the FTO gene risk alleles, other risk alleles, and then metabolic efficiency 3. Well, each of them can have an effect on your risk of a higher weight, but it's remarkably characterized by small effect size. There may be an exception to that, of course, but in the general population, the common form of obesity is typically anchored in small size. So the big trend is small effect size, multiple causes, but most of them with small effect size. And I submit that even though the energy balance equation remains the cornerstone, individuality at the genetic, metabolic, and physiological level accounts for some of the differences in the predisposition to obesity. Thank you for your attention.